Hey guys, it's me Mohammed from the Soft Learners, and in this video we are going to study in detail the G-protein coupled receptors. G-protein coupled receptors. Now there are many types of G-protein coupled receptors like G-stimulatory, G-inhibitory, GQ, G0, and so on. But we will discuss only these four types of receptors. Now starting from the G-stimulatory, consider this our cell. Now this is our cell, and there is a serpentine receptor attached on the cell membrane. Now this serpentine receptor has an external domain and an internal domain. This, as you can see, this receptor has an external domain, ligand binding domain, and an internal domain called as effector domain. Suppose this cell is a myocardial cell, so the receptors present here are beta adrenergic receptors. The receptors present here should be beta adrenergic. Beta adrenergic receptors. Now this class must be present here. Suppose a ligand like epinephrine is going to attach on this ligand binding domain, and the effector domain in response will show us some intracellular signaling. Now as we already know that there is a protein attached, present, coupled here, called as G-protein. G-protein has 3 subunits. Alpha subunit, beta subunit, and gamma subunit. Now this protein has 3 subunits, and there is an GTP is attached with this subunit. When there is no ligand, the GTP molecule will be attached with the alpha nephrin, and it in this state is called as inactive state. But whenever a ligand like epinephrine will attach on this ligand binding domain, this effector domain will go under some transformational changes, and this transformational changes will result in the displacement of this GDP by a GTP. This GDP is displaced by a GTP. Whenever this GTP will attach with this alpha subunit, this alpha subunit will go under some transformational changes. It becomes activated, and it detaches this beta subunit and gamma subunits with itself. Now these beta and gamma subunits are detached from this alpha subunit. Here are our beta subunit, and here are gamma subunit, and this alpha subunit will become activated. Now it is activated. Now this activated alpha subunit along with its GTP will go to another enzyme that is also present on the cell membrane. This enzyme here, it is present in the cell membrane. This activated alpha subunit will go to this enzyme, and attach to a specific site here for the allocation of the alpha subunit. Now this is a specific site where alpha subunit will attach along with its attached GTP. Now this GTP is attached here. Let me show attached GTP here. Here is our alpha subunit. Here is our GTP attached here. Now, whenever alpha subunit along with its attached GTP is attached with this enzyme, these enzymes result in some conformational changes into the intracellular environment. The name of this specific enzyme is adenyl cyclase. So far what we have discussed, that in the inactive state, alpha, beta, and gamma subunits are attached along with the GDP molecule, but whenever a ligand will attach, the effector domain will show some intrinsic changes and the GDP molecule is replaced by a GTP. Beta gamma subunits are displaced, and this activated alpha subunit along with its GTP will go to an enzyme present in the cell membrane. This enzyme is called adenyl cyclase. Now, this adenyl cyclase enzyme will do some transformational changes into the internal cellular environment. And what are these transformational changes? These transformational changes include the conversion of ATP into cyclic AMP. Adenyl cyclase will convert the ATP present in the cell into cyclic AMP. As we can see, epinephrine attaches here as a ligand. This ligand activated an alpha subunit, and alpha subunit goes to the adenyl cyclase and converted and activate adenyl cyclase. Adenyl cyclase in response convert ATP into cyclic AMP. Now this cyclic AMP that is present inside the cell, this cyclic AMP will activate another enzyme that is present in the cell. Here this enzyme is very special, and it has very specific features that changes the transformational changes into the cell. The name of this enzyme is protein kinase A. Cyclic AMP will activate protein kinase A, and protein kinase A will alter many functions in the cell. The alteration includes phosphorylation of many protein present in the cell. So what protein kinase A will do? It will do phosphorylation of many compounds of many proteins present into the cell. For example, it will phosphorylate enzymes. We have enzyme here. Protein kinase A will phosphorylate it, and when our enzymes are phosphorylated, they become active and they alter the metabolism of the cell. Now the first thing they are doing is phosphorylation of the enzymes. The second thing protein kinase A will do is the phosphorylation of many intrinsic factors that is present inside the cell. For example, here we have our intrinsic factor. There we have intrinsic factor present inside the cell. Now this intrinsic factor will, when phosphorylated by the protein kinase A, it is phosphorylated, it become activated, it go into the nucleus and will affect the genetic material. Now what protein kinase A is doing, it is phosphorylating the enzymes which when activated or altering the metabolism present in the cell, metabolism of the cell. Now the second thing they are doing is the phosphorylation of some intrinsic factors which go to the nucleus, and when they go to the nucleus, they alter the genetic material of the cell. The third thing that protein kinase A is doing is the phosphorylation of some protein channels. Now there are protein channels present here. Now there are very special type of protein channels which are operated by calcium. They are calcium operated protein channel. When protein kinase A phosphorylate it, these channels open, calcium will rush into the cell and alter the potential of the cell and will alter the electric potential of the cell. As we can see, G-protein, what are G-protein coupled receptors? They are serpentine receptors linked with a protein trimeric protein which consists of three subunits: alpha subunit, beta subunit, and gamma subunit. Then when there is no ligand attached, alpha, beta, and gamma subunits are attached with each other. But whenever ligand will attach, the GDP attached with the alpha subunit is replaced by a GTP. Beta sub and gamma subunits are detached from the alpha subunit. The alpha subunit will go to an enzyme called adenyl cyclase and activate it. Adenyl cyclase in return will convert ATP into cyclic AMP. Now this cyclic AMP will activate protein kinase A. This protein kinase A result in the phosphorylation of the many components present in the cell. First, it will phosphorylate enzymes. Second, it will phosphorylate many intrinsic factors present in the cell. Third, it will phosphorylate some channels, ion channels that is present in the cell. As we can see, a single ligand will attach. This single ligand will attach in the activation of many G proteins. Now these many G proteins will activate many adenyl cyclases, and these adenyl cyclases will result in the generation of thousands of cyclic AMP, which result in the activation of thousands of protein kinase A. So our signal is being amplified here. A single ligand will result in the activation of many protein kinases A. This system is also called as biological amplifier system. Biological amplifier system. Let me repeat myself. These are the serpentine receptors attached with the G protein. A ligand will attach here. The G protein will activate it. The alpha subunit is activated by the GTP. Beta gamma subunit attached. This activated alpha subunit along with its attached GTP will go to an enzyme called adenyl cyclase. Adenyl cyclase will convert the ATP present in the cell into cyclic AMP. This cyclic AMP will result in the activation of an enzyme called protein kinase A. This protein kinase A will phosphorylate many components. Some are enzymes, some are intrinsic factors, some are protein channels present into the cell. As we can see, whenever a ligand will attach, alpha subunit gets stimulated, and this stimulated subunit will result in the increase of the cyclic AMP. So this protein will specifically called alpha stimulatory, beta stimulatory, gamma stimulatory. So this type of stimulatory G proteins are called GS. These are called GS protein. Now this is all from this video, and in the next video I'm going to show you how this G stimulatory is counter-regulated by another protein called as G inhibitory. Now in the next video we will discuss G inhibitory proteins. This is all from this video. Thanks for watching. Please subscribe our channel.